How did that win feel different than, I mean, in your years at Novartis and Amgen, you played a role in a lot of wins, for sure. How did the win at Atara feel different and perhaps um, influence what you did next, like what you chose to, where you chose to take your career after that,
Yeah,
if at all?
yeah, absolutely. No, it absolutely did. So I would say drug development in general is hard, right? People need to know this is a hard business. It is high risk. It's a very long cycle time. It's difficult. Even more difficult is to try to do something that is entirely new, right? Develop a therapy that is an entirely new modality. It's got all kinds of risks. Lots of discussions with health authorities. What's the regulatory path?
Right.
How do we gauge quality manufacturing for this thing? What kind of data
How
are
do
we
we fund this thing that no
how do
one's
we
ever
fund this thing?
seen before?
Exactly. All of this, right? It's so I think different and particularly exciting and meaningful is, yeah, this company, this team were the first to really make this happen. And it was not easy. It was incredibly challenging. There were many, hurdles and challenges along the way. But it eventually got there, and I'm confident that today there are people alive, enjoying their lives and with their families who otherwise they would not be so, were it not for those efforts.
Mhm.
So, I think um, that's particularly meaningful. That's something that I'll always take with me. And how did that impact me in terms of, preparing me for eventually coming to Poseida? Well, in a few ways. I think I would say first, it gave me absolute conviction that the future of cell therapy must be allogeneic. Um, I just gave you a great example. There are patients out there in a number of diseases who cannot wait for bespoke autologous cell therapies to be manufactured, which can take weeks to months if the capacity is even available, to manufacture that therapy for them. And that's just not acceptable. Cell therapy is so powerful. We need to, offer every patient who deserves to have the benefit of cell therapy to have that opportunity to do so. And for that, we need allogeneic cell therapy that uses great starting material, healthy donor cells as starting material, can be manufactured at scale at much lower cost, and can be, either stored at hospitals or shipped overnight to be ready right when that physician and patient together make the decision that cell therapy is the right next step. So it gave me that absolute conviction. And it also put me in the position of really coming to understand, let's say, the state of play of cell therapy. There are so many cell therapy companies out there, and it can be difficult to really understand how they're different, right? Everybody is making big claims. We're very different from everybody else, but it can be quite challenging, even for people in the field, to understand exactly how.
Mhm.
So since I was in the space, it really gave me the opportunity to go deep and, and learn about all of the companies in the space. And I saw data from Poseida at an ASCO GU conference, genitourinary conference, for a prostate cancer program that Poseida had. And this is a solid tumor. CAR Ts have really struggled in solid tumors. And I saw that data. The poster didn't get very much attention at the time, but I saw it and said, I think that's really remarkable data in solid tumors for CAR T. I'm going to take more of a look under the hood, which I did, and, really learning more about Poseida's technology, I came to the conclusion that Poseida really has the winning set of proprietary, totally different genetic engineering tools and supporting technologies. And so really has all the elements that are necessary to make allogeneic cell therapy happen. And so that started the process by which I ultimately joined Poseida.
Yeah. And when you joined, that was 22, right? Correct? In 22?
It was April of last year, so April of 2023.
Of 23. Okay. Yeah. And you joined as president of the cell therapy division.
Yes, I did.
Yeah.
Yes.
Um, what sort of differentiated that role from the role that you're in now?
Ah, yes. Great question. So Poseida at its core is actually a platform technology company. So you mentioned, you know, Eric Ostertag. So Eric was the first gene therapy student out of the University of Pennsylvania, very first one.
Hmm.
And way back then, he quickly came to the conclusion that the kind of tools that were available, a lot of which relied on viral vector technology, just would never be able to deliver the profiles of the kinds of medicines that we would ultimately need. So a lot of limitations of viral technology. So there's a lot of safety concerns, expensive and difficult to manufacture. But also the versatility is limited. You can only pack so much DNA in a virus. And so it's very difficult to put a lot of functionality into your medicine. So he developed an entirely different set of tools and completely unique approaches to do this. So we use those tools today on two sides of the business. So we use them in an ex vivo or outside the body way to engineer cells, and that is our allogeneic CAR T. But we can also use them in vivo as genetic medicines. And so we have a program in gene editing, for example, a program in non-viral gene insertion. So I came in to start leading just the cell therapy, the allogeneic cell therapy division, as you said, but Poseida actually has much more, many more programs and much more technology under its umbrella than just cell therapy, though those programs are in the lead.
Yeah, yeah. And I mean, that sort of begs the question, given so Poseida being a cell therapy company, a gene therapy company, and even within the cell therapy program, like the solid tumor programs, it's a pretty broad array of indications that you're looking at, right? I mean, solid tumors across multiple indications. Um, how would you say your experiences, from McKinsey through Big Bio, sort of prepared you to, move from a cell therapy discipline, leadership of a very specific cell therapy discipline, to leadership of a company that is multifaceted, to say the least, in terms of the targets and indications it's going
Yeah.
after?
Well, so I would say I am really grateful for all the preparation and all the training that I had, starting with consulting, being at Novartis and Amgen, because I really did have the opportunity to do just about everything. So I was in many commercial roles, global, in country, pipeline, in market, lifecycle management, but also time in clinical development, strategy, business development. I was originally trained as a biochemical engineer. So, what do? You culture cells. So, it's been, definitely something of a wandering career journey. I would be the first to admit that, but I'm very grateful to have had all the different experiences in different functions and seeing different things that I have, because I think that has really given me a great perspective on all the different areas, that need to happen, all the different capabilities a company needs to be successful. I mentioned corporate communications at Atara. That taught me about investor relations and managing the whole investor side. So all of these different experiences, and I wouldn't say that someone has to have all of them in order to take on a leadership role like CEO. But I will say I feel very well prepared, having seen, and played a role in so many different places. And I think that helps me be calibrated on, what's likely to be an issue that needs real attention, maybe needs my personal attention, versus other things that are just going to work themselves out, or it's just, kind of business as usual. Yeah, it's a challenge, but we're going to work through it. And that, I think, is a very important and valuable skill, because no one can be everywhere at once, right? You really need to pick your spots, and you need to pick your altitude. And again, I'm very thankful that I had, so much opportunity to see so many things and so many different therapies in development or on the market or whatever, because it really gives me comfort and confidence in knowing where to go in that way.
Yeah, yeah. When you took on the CEO role, what led to that? Like, what was the sequence of events? There was some transition there from Eric to was Mark Gergen
Yes,
the
absolutely.
CEO
Yes,
in between the two of
yes.
you? Then I always hesitate to ask these questions about CEO transitions because sometimes there's uncomfortable stories behind those transitions. But what led you from president of the cell therapy practice to CEO of the whole company?
Yeah. Well, I think this was all very well planned. So one of the things I love about Poseida is very systematic. For a small company, we really try to be, rigorous and move quickly, but be, you know, have some method, and not just madness all the time. So,
Mm.
you're absolutely right. So Eric founded the company, really put in place what we have that's Poseida today. But then around the time of the first transition, which was moving the programs, from being a research stage company to starting to go into the clinic, that's where the transition to Mark Gergen came. And Mark, who still currently serves as our executive chairman, by the way, had an absolutely fantastic set of skills and experiences for the company at its next stage. He is a phenomenal deal maker and, just has a legendary resume within biotech. Amongst other things, he was one of the co-founders of Mirati. So he was really instrumental in providing the foundation for Poseida in the form of some of these partnerships, capitalizing the company. So, putting together the Roche deal, helping to orchestrate the first deal with Astellas. I was just looking back, actually earlier this week, at the non-dilutive capital that the company has raised. We've only ever done one small equity raise since the time of the IPO. And over the last 3 years alone, we've secured $400 million in non-dilutive financing through these partnerships. And we've got, maybe up to another 150 million coming potentially just in the next 12 to 18 months alone. So Mark, really put all of that in place for Poseida, and then saw that the next transition point was coming, the next inflection point, which is for Poseida to be, really a full-fledged clinical stage company, which we are. We have 3 programs in the clinic. We have, several programs rapidly approaching the clinic in the CAR T, and then we have, the genetic medicines programs, and we've said we'll have at least one IND in 2025 on that side as well. So, for Poseida now in our next phase, this is where the rubber hits the road in execution of clinical development, strategy, delivering data, thinking about target product profiles, making sure we have the data to support them, all of that. And I think that's, where my experience and my skill set, and having seen an allogeneic T cell immunotherapy all the way across the finish line,
Mhm.
those are some of the skills that we're looking for now.
Yeah, I mean, you've got some, as we've ascertained, some commercial chops. So I don't want to get too far ahead of our skis here, but I mean, I imagine putting Dr. Kristen Urema in this position puts you in a nice position to stay there at least for some. I don't want to say long term. We don't want to look too far into the future, but for some amount of time, given your commercial chops.
I could not be happier at Poseida. This is right where I want to be. Poseida is the real deal. I truly believe we are probably the emerging leader in allogeneic cell therapy and genetic medicines.
Yeah.
You know, and it's the technology, and it is, like I said earlier, equally the people. You know, the culture here is very, very special. You know, people have a really unique ability to just make things happen. And I've seen a lot of companies. I feel, you know, very, very well calibrated on that. I tell our people that all the time. I'm like, you don't understand how special this place really is. You know, it's just look around you, look at your colleagues, you know, be thankful for them.
Yeah, that's great. Give us a little update on how the pipeline has kind of come together in the time that you've been there and shortly before, an update on that. And then I want to talk a little bit more specifically about some of the partnership deals because I mean that's so important. You know, not just from a product development standpoint, moving the ball down the field, but to your point, I mean it's been a financial boon to Poseida as well, right? Keeping the lights on, keeping the employees getting their paychecks
Absolutely.
and doing the good work. So, so let's start with the pipeline though.
Yeah.
Just give us an update on recent pipeline advance.
Absolutely. So the pipeline is going like gangbusters. I guess that's it in a nutshell. So, you know, it really started, I would say, with ASH last December of 2023. So that was the first time that we really put up a data set for the program that is the most in the lead, which is the myeloma program, PBCMA Allo1. So this is one of the two programs, heme programs, that's partnered with Roche, the lead in that partnership. And it was an early data set, but what we showed was that an allogeneic CAR T really has the promise to be at least as good as an autologous CAR T. And I think right now that's something that most of the industry does not believe, right? There's been a lot of challenges in allogeneic CAR T, you know, a lot of there's a lot of hype and some setbacks. We believe that you need a particular kind of allogeneic T cell to be the foundation of your CAR T, the T stem cell memory cell. Not just any old T cell will do. It's got to be that. And to get that kind of CAR T, you can't use a viral technology. You really have to use a transposon technology, which is what we use. So this was the first data set for a program, you know, with that profile. And what we showed is in a very, very heavily pretreated population, probably the sickest multiple myeloma population ever treated, we saw an overall response rate that was, you know, incredibly competitive with the autologous CAR Ts. And we saw great depth of response. It was a very early data set, so we didn't really know about durability at that time, but we really liked the leading indicators of what we saw. We could see our cells. We measured them by flow cytometry. We could see them sticking around in the bone marrow where they're supposed to be, fighting the tumor. So we're incredibly encouraged. You know, our partner Roche also incredibly encouraged. And so that program has been continuing ever since. And what we said is we'll give at least one data update, possibly multiple data updates on that program later this year, in the second half of this year. And it's been running now for, you know, in the kind of doses and cohorts that are probably of most interest for over a year. So by the time we give a data update on this, you know, it's going to be a very meaty one. And I think we are going to surprise the world with, you know, hopefully a really, really interesting set of data that shows, you know, an off-the-shelf allogeneic CAR T that patients do not have to wait for, that everybody can receive, and that can be manufactured at scale. We're getting 100 doses from a single patient, single batch right now, depending on what you believe about the cell dose. You know, this is not a dream. This is, you know, this is coming. So that's, you know, really put wind in our sails, as you can imagine. But it's not the only program that we have in the clinic. So we also have a second heme program. This is a dual CAR T. So sometimes we call it dual CAR T. It's really a tandem. It just has domains on one CAR that bind both proteins. This is not that. We have two full-length CARs in our cell, one against CD19 and one against CD20. And those are both great targets for B-cell malignancies like non-Hodgkin's lymphoma. So this is a program that we think would be able to be used in patients who've failed one of the commercial CD19 CAR Ts, because a lot of times that's due to antigen escape. They lose the CD19. Obviously, your CAR T doesn't work anymore, but they'll have CD20. Or you could even use it before, and it's kind of an insurance policy if the patient's tumor evolves and they do lose, the CD19 over time. So that program is enrolling. We'll give a data update. We haven't released any data on that program yet, but we'll give a data update on that one this year as well. And then, as you mentioned, we have our solid tumor program. This is against mucin 1C, which is a target expressed only on cancer cells in a range of epithelial cell malignancies. So that program, we continue to enroll. We'll give a data update there also. And most recently on that program, we had a poster at AACR where we showed in solid tumors, we've always known that solid tumors are a more difficult nut to crack with CAR T than heme malignancies. We think maybe one of the reasons why is it's more difficult to get an adequate conditioning, what we call lymphodepletion of those patients, which is what you need. You kind of need to clear out some of the immune system cells to make space or niche for the CAR T cells to expand, differentiate, become the tumor killing cells that you're looking for. And so what we showed is if you go to a higher level of lymphodepletion, we can get much better expansion of the cells. So we're very excited with this new learning about, what we will be seeing in that program. And again, we'll give a data update there. So all of that is just on the cell therapy side. At ASGCT, we show that our lead gene editing program, which is for hereditary angioedema, that our gene editor is probably 20 to 40 times higher fidelity than Cas-CLOVER, which is what everybody's excited about already. So, really promising technology there. And we also have a non-viral program, fully non-viral program, that's a gene therapy for factor VIII, for hemophilia A. That's an area that's, had some challenges with gene therapy in the past, but we think those are because those were viral gene therapy programs, not non-viral gene therapy programs. And a problem that those programs have had is you don't get durable expression of the factor VIII protein, which is what you're looking for when you're treating hemophilia. Well, we showed, and this was in rodents, but we'll be moving to primates, non-human primates, we showed rock steady expression of the factor VIII protein at therapeutic levels all the way through 13 months. This really surprised, all of the practitioners and thought leaders who were there. So we feel like the pipeline is just, it's like an embarrassment of riches. Everything is moving forward as we would hope. If we had more capital, we could fund more things, but we're focused on those things in the pipeline right now.
Yeah. And the embarrassment of riches has obviously attracted the attention of companies like Roche and Astellas. That being said, you don't just put out some good news and have all comers, knocking down your door. So I'm hoping we can dig in a little bit into more, probably specifically the Astellas, the refresh of the Astellas deal that you've probably played more of a hand in. The Roche deal was kind of pre your time. So, and sort of how it came together. So I guess I'd just start there. Like first, what is the collaboration entail? Like, what does the collaboration look like? And then maybe follow that up with how it came to pass.
Yeah, absolutely. So this is the second deal that we did with Astellas. So there was a first deal which was a little more arm's length. So it was essentially a strategic investment on the part of Astellas into Poseida. So they got some equity, they got a board observer seat, some limited rights of negotiation on a program that were time bound. So it wasn't like a real collaboration. It's still very meaningful, and we appreciated starting that relationship, but we weren't working together. But this spring, we announced, in a way that Astellas is doubling down on Poseida, if you will, by announcing a real collaboration and license agreement. And we are really excited about this because I think it's an example of where, it's not just a license of something from one company to another. This is really bringing together technology from two companies that's incredibly complementary and creates the potential for new medicines that neither company would have been able to deliver on alone. So specifically, this is regarding a convertible CAR T. Well, what the heck is that? I like to think of it as, there's the screwdrivers with the exchangeable tips. So if you think about the handle of the screwdriver, that's like the CAR carrying cell itself. When you've got a CAR T, that's the long part of the screwdrivers, sticking up with some binder on the end. And then the convertible tips are, what you swap in. So Astellas has a technology through their subsidiary Zyphos that they call MicAbodies that are these interchangeable tips. So what the arrangement is Poseida will produce that chassis car, that handle of the screwdriver. And then Poseida or Astellas rather, will provide up to 2 of those interchangeable tips. And what's really cool about this technology is, you can, it has manufacturing advantages, right? Maybe you manufacture the cell once and then you're just essentially manufacturing an antibody component. So that probably has some cost advantages, maybe some regulatory advantages. But it also opens up the possibility to go after different targets or maybe even to change targets in real time. So you could, in theory, by mass action in a patient, give them the CAR T cell, give them an initial MicAbody, then maybe infuse a different MicAbody later, knock off the first one, and then you've kind of changed the target. Or you could give a mix of MicAbodies, and then maybe you have a multifunctional cell with CAR Ts that are, directed at different targets. So this is not an approach that Poseida was, planning to pursue. We weren't looking at a convertible CAR technology. And for their part, Astellas, didn't have, I think they thought, the right type of cell. Remember, I mentioned not just any CAR T will do, you really want the T stem cell memory cell. So if so we're providing the allogeneic T stem cell memory CAR T, and they're providing their convertible CAR tips, these MicAbodies. And so this is in solid tumors. And opening up space that neither company would have been able to pursue independently. So it's really, I think, a great example of a win-win.
Yeah, you mentioned that that it doubled down on the relationship, and I'm curious, and you're actually working together now. Like, I'm curious about what that looks like in practice. Like, what is, because these partnerships take on just so many different permutations and form factors. What does working with them look like? I mean, are your people like in constant contact? Are they, sleeves rolled up in the lab with their people? What does it kind of look like?
Yeah, absolutely. So, what's great about the two companies is I think they share, really a high science, set of values and culture, along with a passion for patients. I would say the same is true, of our partners at Roche too. And that's very important because, it means that the scientists get very excited to talk to each other, to work together. So, even in the run up to this, there was a lot of excitement. And when we finally signed it and started to get going, like from day one, the scientists are talking together, they're meeting, they're charting out the plans, working on selecting the targets. So it really is a very hands-on, very fluid communication, sort of a setup. And I think that's really important if you want to maximize speed. If you're kind of shuttling communications back and forth, which is another model, you can do that, but you're going to lose a lot of time. So yeah. I think it was really easy to get into that mode of working very well together and constant communication because we already had that previous relationship. So like I said, it wasn't an active collaboration, but we did talk to them all the time. We had standing meetings, regularly once a month. So there was already a lot of I would say, understanding of the companies mutually, a lot of trust, a lot of respect. And so that just, really kickstarted, I would say, our ability to, to work immediately out of the gate once we entered this new phase.